Hello, everybody. Thank you for joining us for a special look at what our restoration teams are doing on a ship in the deep Gulf. My name is Sasha Francis, and I'm the Restoration Education and Outreach Manager, and I will be your host today. So we'll get to talk to our friends on the RV Point Sur, which is a 135-foot vessel owned by the University of Southern Mississippi and operated by the Louisiana University's Marine Consortium, or LUNCON. Today, the team is about 60 miles off the coast of Louisiana. And throughout today's program, please share any questions or comments that come up in the chat. If you're watching a recording of this live stream, listen in at the end for Q&A and check out links in the description to learn more about these projects. And we're going to pause for folks tuning in live for questions as we go. All right. So we'd love to get your feedback on today's program as well. So we have a short survey you can fill out to tell us a little bit about your experience. So we'll send that link to the chat now and again at the end of the program. We would love to have you fill that out before you log off today. So please check it out. All right. In just a few minutes, we'll connect with folks on board. But first, we're going to talk about where they are and why they're out there doing this work in the Gulf. So let's check out a little overview video of some great footage that we have here. So as you travel deeper in the ocean, less and less sunlight is able to penetrate the water. Just past recreational scuba diving limits, you'll reach the mesophotic zone, which means middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, fish, and invertebrates. And beyond this twilight zone, there's no sunlight at all, as well as colder temperatures and higher pressure. But there are still vibrant habitats full of life at these depths, hundreds to thousands of feet below the surface. These mesophotic and deep benthic or seafloor ecosystems are found worldwide, including in the Gulf, which is actually where all this awesome footage is from. However, the 2010 Deepwater Horizon oil spill dealt a heavy blow to Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted by this spill. Over 770 square miles of deep sea habitat were impacted by the spill. And as a result, collaborative restoration projects are underway to help us understand and restore these seafloor ecosystems found below 160 feet deep. These projects are developing new and improved maps of the seafloor to locate these important coral communities, learning more about the species that live in these habitats, the environmental conditions they thrive in, and how they may be connected across the Gulf. They're determining the best methods to encourage deep sea coral growth and reproduction to help restore impacted coral species and the important habitat they create, and reducing threats that these ecosystems face. They're also connecting the habitats and restoration work to the public and natural resource managers through programs like this. This restoration work, made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees, is led by the National Oceanic and Atmospheric Administration and Department of the Interior, plus many partners, including a large number of academic institutions and non-governmental organizations. So hopefully you got a little bit of an overview of the different restoration projects we're going to be talking about today. And now we're going to connect with the ship to give you some more things to learn today, now that you've had that overview. So let's see who we have on board today. Hello, everyone.
Hello.
How's it going? Is that an octopus on the microphone?
Yes, we are 3.5.
3.5 octopus. Okay. Don't worry. It's just a stuffed octopus. We don't have a half real octopus on board. All right. Well, let's see. I'd love for y'all to introduce yourselves, since I haven't met a couple of you in person. If you can introduce yourselves and tell the folks here tuning in what you're doing on board, maybe what your specialty is, and also what university you work for, or what institution that you work for, or organization you work for.
Yeah. So I can start. So I am. My name is Jordan Hyman. And I am a postdoctoral research science for the University of Southern Mississippi. So the people who own our lovely RV Point Sur that we're on today. And I study the microbial communities. So those are our bacteria and archaea that are living in our deep sea sediments.
Hi, I am Elisa Baldrighi. I am a research assistant professor. I come from the University of Nevada, Reno. And my specialties are a tiny organism called myofauna. They are organism of 30, 40 micrometer, more or less in size. And they are a lot, hyper diverse, and then they live into the sediment.
Hello, my name is Caroline, and I work with U.S. Geological Survey. I work in a benthic ecology laboratory, and we look at all of the critters that we find in the mud.
Awesome. I like that you're in size order of like the creatures that you work with. So we can have, bigger ones and then still pretty small, right? In the middle, and then really tiny need a microscope for.
Yeah.
All right, cool. And I think we'll have some cool footage of kind of all those varieties as we go today. But I wanted to start out by asking, how do we collect samples of mud? Like, what's it? I heard the word multi-core, and tell me about the different tools you use. How do they work?
Yeah, so we are using a multi-core. The one that we're using today is called a mega core, actually. And so it is, as you see right in the footage here, so it plunges into the ocean with those clear tubes that you're seeing. And that clear tube then collects our sediment. And that sediment in that tube is actually going to represent the seafloor as it is basically sitting at the bottom of the ocean. So it'll keep any sort of layering that's occurring at the bottom of the ocean so that when we get it back here on the ship, it's representative of what's actually occurring and it hasn't changed too much on its way up. And we take those cores out of those black boxes that you're seeing in this video here. And that is super important for us to be able to, like I said, get a representative sample of what is actually happening at the seafloor instead of changing things on its way up, because it's a long way for it to travel.
Wow, that's really cool. That's pretty impressive that it can come up. How deep, for example, are you doing cores on this mission to give folks an idea of like how far that has to travel to not have the mud fall out?
Yeah, so many of our sites for this trip are roughly 1,000 meters to about 1,500 meters.
Wow, so that's over 3,000 feet. Okay,
Yeah.
Incredible. So can you tell us a little bit about the journey of a sample? So we've got the core, we get the mud, we bring it up. We saw a little bit of how they kind of process that or at least kind of get it off the multi-core. And then what happens? Like what's everybody's job kind of once that comes on deck?
So what we do, I can start talking about myofauna. What we do, we insert a smaller core into the big core. Sorry, we subsample the big core with a smaller core, and then we bring the smaller core inside the wet lab, and then we slice the first 3 centimeter one by one, and then we fix our sediment into the formalin, and we ship the sediment into the lab. Unfortunately, our animals are so tiny that we cannot see anything when we are on board. So we have to come back to the lab and analyze our sample under the microscope. So this is what we do for the myofauna.
And then for the macrofauna team, we essentially bring our cores inside and we slice them up into 5 fractions. For sediment chemistry, which is a little bit different, we use 6 fractions. And then we put them in containers called Nalgenes that the sample goes into. And then when we get back to the lab, we run our sample through a sieve. So we basically are just left with the macrofauna level. And some of our critters you can actually see with your eyes, which is really cool.
Yeah, they are.
And then we look at everything underneath a microscope, and we take note and we collect the creatures that we find.
Yeah, and then so for the microbial team, you can actually see that happening live right now. And so that we actually take out of the big cores that come directly off of our mega core, and we sample all the way down to 10 centimeters. And we actually cannot see our creatures because they are microscopic, they're microbial. And so we actually never get to look at them either under a microscope. They're that small, and we can get a better representation of our microbial communities via sequencing. So we actually take our mud sample back to our lab and then extract DNA and sequence that DNA. And that can actually tell us what species of bacteria and archaea are living in our mud at a much better representation than if we looked at them on a slide, for example.
So it sounds like y'all have a lot of surprises when you get home. So
Yeah.
you can do the work collecting all the mud, but then really you don't really know what you're going to get right till you get back. So it's like a fun surprise, right?
Yeah.
Hopefully fun surprises and good things that you were looking for, maybe something fun you didn't expect. So as far as launching the core and the other things, are there any other kind of roles on board that have to help with the sampling? And then once you know, get it back on board, kind of packing it all up, are there any other roles that you'd like to mention that are part of that?
Yeah, so I can talk about the video that's happening right now. So this is the deployment of the mega core. And so it's super important when we are on board that everyone works really well as a team to get our equipment off of the ship in a smooth way, and that our mega core doesn't break or hit the bottom of the seafloor too hard or things like that. And our crew does a really, really great job of keeping everyone safe and making sure that everyone is on the same page. And we slowly move that core off of the ship. And so that deployment, and then we do sort of basically the reverse of what you guys watched for the recovery and bring it back on ship in order to get those cores full of mud off.
Very cool. And I also imagine that the ship crew, the captain and folks driving the ship have to kind of keep it in position when you're doing that launch too. So there's a lot of teamwork that goes into that.
Yeah,
Awesome.
super critical.
Oh, yeah.
So what do you find in the mud? Like what's your kind of favorite thing that you've seen while processing a sample? What other things do you see when you're looking through the mud?
So my most memorable moment that I've had with a core, actually, it's been on board the Pointster. It was one of my first mud cruises that I went on. There was a cumation that was actually swimming up in the core tube, and I thought that was really neat. I had seen cumations before, but I had never seen them actually swimming, and it was adorable, and I loved it so much. And then there are also some of my favorites are the crustaceans. So like taneids and isopods. I love them so much. I think they're so cute.
Well, since I cannot see my tiny organism, so I usually spy all the time what she's doing, and then also from the macrofauna core, we can say I remember seeing beautiful worms.
Yep.
Called polychaetes. They usually are very abundant also in
Yeah.
your samples, and then they are just fantastic. In the past, I work a lot with the macrofauna, so it's beautiful every time digging in their cores and then see these animals still alive and coming up from so deep and from the ocean actually.
Yeah, I'm gonna agree with Elisa on this one. Yeah, we can't see the bacteria, but we do get some really cool macrofauna, and we all have an appreciation for each other's communities as well. And so visibly, even though if I can't see my communities, I do get really cool data about my communities, but visually, yeah, seeing some of the really cool worms kind of travel their way through our cores is always very exciting.
Oh yeah, and we're seeing some on screen right now.
Yeah.
Some of the stuff you might not be able to see right away, but you might get to see later on, which is really cool. So what do the critters in the mud tell us about the Gulf ecosystem? Like how are these things connected? What are we learning from finding these things?
So I can answer to this question. So basically, sediments and the organism that live into the sediment are an excellent memories of what happened in the past and what is going on now. So every changes, even if it is minimum changes, is registered into the diversity and into the abundance of these organisms. So basically, what my organism can tell Jordan organism, microorganism can tell a macrofauna is if the system is healthy or not, if it is suffering or not, if there is any kind of pollution, and if actually the recovery is going on or not. So they can tell us everything, what happened and what is going on. So again, it's a good memory, it's a reliable memory of everything that is going on down there. That's what I can say. That's my definition. I don't know if you want to add something, girls.
Yeah, I mean, kind of like Elisa said, it's all connected. Macro fauna depend on myofauna, which depend on microbial communities. And so we get a lot of really cool information looking at all three different levels of this community with this team that we have here.
Yeah. I can add that studying the sediments, we can add, and the animals that live into the sediments, and bacteria and prokaryotes, microbes, we can have extremely important information on the functioning of the system. This difficult word is a word to say how the ecosystem is working and how everything is connected.
Very cool. And so, as far as how you're helping with the restoration work here, how does all of that kind of inform what we're doing to restore the Gulf or help with that restoration work?
Yeah, so like Elisa talked about with the health of the ecosystem, you can't really establish a successful restoration program without understanding the ecosystem's health before you start that program. And so we can establish a lot of ecosystem health and whether that ecosystem is recovering, especially related to that Deepwater Horizon oil spill that MDBC is really interested in. And so restoration is really dependent on that ecosystem health to start that restoration project. And so our communities really create that base of ecosystem health and understanding how that health of that ecosystem is potentially changing, and that could be related to a restoration project.
Excellent. Yeah, we really can't restore really well unless we know what's down there, right? Unless we know the
Yeah.
whole picture of these habitats, and knowing all of the tiny things is a really important part of that big picture, right?
Yeah.
Awesome. Fantastic. So, I wanted to maybe take a moment to see if we have any questions from the audience yet. And we'll see if we can pull some of those questions up. And while we're doing that, in the meantime, do y'all want to maybe mention how you got into this work? If you want to kind of go down the line, and then we'll take a little break for questions as they pop in.
Sure. I can go for it.
Yep.
So I got my bachelor's of science at UNF, and then I actually was a park ranger for a year, and that was a temporary position. And I found this one, and I applied to it, and my boss gave me a call, and I was really, really lucky. I was one of the ones that was picked for another interview, and after the interview process, I was accepted into the position, and I really love it. I didn't know anything about mud or the creatures that live in the mud, and I have learned so much, and I have so much appreciation for mud. It's the basis of the ecosystem in the ocean. Like, if the creatures and the microbes and everything in the mud is not healthy, then the ecosystem is not going to be healthy and thriving throughout the whole water column. So it's pretty neat.
So I come from Italy. That's clear that I'm not American from my accent. So I study marine biology in Italy, and I got my PhD. And so my boss, there is a professor at the UNR, reached out to me with an email. He got my contact from a dear colleague of mine in Italy. So this is how the networking is so important in our job. So he reached out to me, sent me an email, and say, "Well, listen, I'm looking for someone that is expert in myofauna, especially nematodes. There are little worms that belong to the myofauna. And then I have this big project in the Gulf. And are you interested in?" And I say, "Oh, yeah, absolutely, because I am a marine biologist and then deep sea ecologist. So this is my specialty." So we communicate like a couple of emails. I got an interview, and then he said, "Well, listen, Elisa, if you want, the job is yours." Yes, of course. So I got the visa and I flew to Reno in 2023. So it has been like, this is my third year being part of this project, that it's just amazing. Yep.
Yeah, so I got my bachelor's in just biology in Missouri. So I was super landlocked, and I always kind of wanted to make my way to the ocean. And I ended up doing my PhD in freshwater and marine microbial ecology. And so that sort of got me started in marine work. And through one of the grants that I was working on at that time, I met my current supervisor, and she and I got to talking, and we had a lot of similar research interests and things like that. And so she actually had this position open specifically to work on this project and analyze a lot of the data that we had collected over the last several years, and get a lot of publications out and things like that organized and ready to go. And so I was super happy to join this lab and join this project and this wonderful team working on such a broad diversity of communities, which is pretty rare in my opinion.
That's amazing. And have all of you been on the Point Sur before?
Yes. Yes.
Oh, okay. So you're seasoned experts at this this particular vessel and everything that you need to do on board for this vessel, huh?
Yeah, we really love this vessel. I mean, I'm a little biased being from USM, but
It's a lovely
It's a very
vessel.
great
It's
boat.
like, like to come back to a big family every
Yeah.
time.
Exactly. Yeah, I love this vessel. The crew is
Yeah.
incredible. The
Yeah.
science team is absolutely amazing.
Yeah.
We're all really close.
Yeah.
We work together really well.
Yeah, there is a good feeling, good collaboration,
Yep.
and then everybody is so proactive. It is extremely important because we work many hours every day. We wake up very early. We work hard. So, and then if you can feel the collaboration coming from the team, that's the most important thing, I guess.
Yeah. And as a quick aside, since you're talking about how much you love the boat, what's your favorite thing you've eaten on this trip so far?
Oh,
Listen, Chef Lawrence is absolutely incredible.
Yes.
Almost everything literally everything he makes is amazing. For breakfast, it's always the bacon. His bacon is perfect. And then, honestly, I've never had anything not good on this ship.
Yeah.
I honestly don't know.
Never. No, that has not
To
happened.
pick a favorite thing is so hard because it's all so delicious.
Yeah.
To be honest with
Yeah.
you, like to be honest with you, it really is. His food is absolutely incredible. I don't know if I can pick honestly.
I'm not
Well, that
sure.
relates.
I agree.
Yeah.
Yeah.
That relates to a question we did have in the chat for what's your favorite part about being at sea? And I'm sure part of it is the food, but did you all want to know any other favorites about being at sea? And then we'll answer a few more questions from the audience.
Yeah, I love just being out on the water. That's kind of one of my favorite things. And you just have really nice views. Really had a beautiful sunrise this morning
Yeah.
while processing some mud cores. But I really just love working and actually seeing these people in person because a lot of times, I mean, you noted we were from all over the country, and so a lot of times we're on Zoom, and so it's just really nice to actually see each other in person, collaborate in person, and work with such an amazing team.
Yeah, I totally agree with you, Jordan. And what I just said to the girls say, I never seen such a beautiful sunrise and sunset like the one that I can appreciate it here from the Gulf. And then, yeah, I would say I would pick sunsets and sunrise definitely, and the peace that you can feel every time that you sail through the ocean. Yeah, I would say that.
Love that.
And then for me, I'm gonna be a third on the views and the collaboration because the sunrises and sunsets and the stargazing is absolutely incredible. I was looking at the stars for about 30 minutes last night when we got done. We got done a little bit late. And then the collaboration is amazing. Actually seeing everyone and like working with people in person and being able to connect in that way is incredible because everyone is amazing. We all work together as a team. And when we come together, we get so much science done. It's amazing.
Awesome. All right, we have a couple more questions. There was one that said, what are the research questions you're trying to answer, or what are your research questions? So I wanted to speak to that a little bit. So this work is a little bit different from a lot of potentially research work that all of you have done in the past. This being restoration work, really all the science we're doing is very specifically to inform and contribute to that restoration of these habitats that were injured by the oil spill. So it's a little bit different in that way. So every single thing we do is to contribute to that overarching mission. So I know we talked a little bit about we can't really restore these habitats until we understand what's there. So I think a lot of the questions y'all are answering with your sediment sampling is what's down there, whether it be what kind of bacteria is living down there, what kind of microorganisms are living down there. That's helping us create that big picture of what is in these habitats that were injured by the oil spill, how are they all connected, and how can we best support their restoration and ensure their resiliency in the future. And then also, these projects are wanting to restore these habitats by restoring the invertebrate and fish abundance for injured species, and then focusing on these, high density mesophotic and deep water coral sites, so that we can kind of create this continuous healthy habitat from the coast to way offshore, and also actively manage these habitats to prevent threats and protect against threats, and provide a framework for monitoring these habitats for education and outreach, like through programs like this, installing mooring buoys for folks to be able to access the open Gulf without, creating any more threats to the habitats, et cetera. So I wanted to make sure I can provide some answers to those questions, but if there's anything else anybody would like to chime in on that, feel free, as you've been working on these projects.
Yeah, I think you covered it really well. I mean, for these groups specifically, we're really looking at, you know, the benthic communities, the three that we're sitting here representing, and how those communities have recovered since Deepwater Horizon, so that we can inform, like you said, those restoration efforts down the line.
Awesome. Oh, and I didn't quite note, but part of this big picture that we're getting by doing the sediment sampling, giving us a big picture of the habitats as a whole, contributes to this overarching restoration. And another thing that we're doing in these habitats is also trying to provide opportunities for new coral growth. And while that might not be something we're focusing on this mission, I wanted to mention that for the folks that are tuning in. Something else we're doing is trying to support propagation or new growth of corals in these areas that were injured by the oil spill, because there was a lot of loss of corals. So that's something else we're doing through different labs that we have, federal labs. There's a USGS lab and 2 NOAA labs, as well as some partner aquariums, where we're actually learning about how the corals grow and how they reproduce, so that we can help actually replace some of the ones that were lost with new small colonies that can continue to grow. So you'll see on screen here just a little shot from one of our coral labs, of those aquarists taking care of them. So talking about the whole picture, that's another cool part of this restoration work. All right, let's see what other questions we have here. Let's see in this chat. All right, did you find and describe any new species? I don't know if we have any answers of that related to sediment sampling per se, but any thoughts or notes on that from y'all?
Well, what can I tell you is that there are so many undescribed species in the Myofauna that I'm pretty sure that our sample are full of new species that need to be described, and this is one of actually of the objective that we have. Describe a new species can take a lot of time. I did it in the past, working on other samples, and it took me a year to describe a species of a nematode. So, well, this is the next step of our research, and that's just preserve what we found, and then eventually describe new species. Yeah, absolutely.
Very cool. I think we might have some articles about describing new species, or kind of that process, or maybe some we have just described during this restoration work. But yeah, it can take a long time, huh? Like what a journey that is, I'm sure.
Yeah.
All right, yeah, looks like we dropped a little bit more info about that in the chat for folks that are tuning in. All right, let's see what else we have here.
Oh, I don't know if
All
I
right.
know the number. Wants to know how heavy the multi-core is. It is, I don't know if I know an exact number, but my guess would be almost 1,000 pounds would probably be my guess.
Wow, that's heavier than I would think.
Very heavy, yes. So it has to be used by it's called a winch, but essentially like a crane operation type deal. And that's operated by our wonderful crew here on the RV Point Sur. And so really when you're watching our videos, we're not picking that up at all. We're really just helping guide it off of the ship, because it's definitely not something that people could lift, even a whole team of us.
Mm-mm.
You're all very strong, but that's a little bit much, I feel like.
It's a little much, yeah.
Yeah.
Yeah. Okay, awesome. All right, I think we might have touched on this in the beginning, but for folks that maybe were a little late to the stream, what is the depth you're taking these samples at?
Yeah, so most of our sites for this trip are around 1,000 meters to about 1,500 meters is one of our deepest sites for this project.
Ooh, that's pretty deep.
So yeah, multiply that by 3 to get feet. So yeah, roughly around 3,000 plus feet, approximately.
All right. And I think that was I just moved my screen, so I hope I didn't miss a question that they pulled up, but it looks like that was the most recent one, right? Yes. Okay. All right. Let's see what else we got here from our chat. All right. So we got the depth here. Oh, here we go. What distance from the epicenter of the spill are you taking samples from? Okay.
Yeah, so we have a range actually of distances. So we have actually a site that is very close to that. It's actually a wreckage of that oil spill. So Deepwater Horizon exploded and then wrecked at the bottom of the ocean. And so we have sites pretty close to that oil spill to really gauge, that higher impact areas. And then we have some of our sites, like, for example, that we're visiting today that are several nautical miles away that were actually considered not impacted at the time of the oil spill. And that really gives us a great comparison for all of our communities to understand, if we're moving closer to that oil spill epicenter, what is some of the differences that we're seeing, or is there a sort of a difference in recovery between the sites that are closer to that oil spill site versus further away?
Awesome. Great. All right. I think we're going to wrap up our audience questions here. And I know that the folks behind the scenes also included some information in the chat to answer some other questions. So let's wrap up with something I've been wanting to ask. So I've worked on these projects for a few years with many amazing scientists like yourselves that work with sediment samples. So I've learned that mud matters, as I like to tell people. And I think we learned a lot from y'all today. But for some folks that are watching that maybe don't have that appreciation yet for mud, why do you care about it? What fascinates you about this work?
You want to start?
Yes, I'll go first. I absolutely love all of the different creatures that we find in the mud. Before I started this job, I had no idea that there was so much diversity in mud. I honestly never really thought about it. When I thought about ecosystems, I usually thought about the bigger animals that we see. And I never really thought about what we see in the mud. And then the different morphology of the different creatures that we see is just so cute. The tanaids are some of my favorite. They have little claws. Yeah, I just love the diversity that we have.
Yeah, that's cool. Yeah, I totally agree with Caroline. And then another thing that fascinated me a lot is like how these animals can live in so deep, no light, high pressure, very little to eat, and then it is extremely cold. So all the physiological and physical adaptation that they have, and how they can thrive in this environment. So if we know a little bit about these animals and then how they live, we can know so much about the environment. So that's just incredible, and they are so tiny and they are so abundant.
Yeah, that's kind of similar to my answer as well. Bacteria are so diverse. Archaea are so microbial communities in general are so diverse. They're actually probably the most diverse out of our 3 communities. Not probably, definitely.
Definitely.
And I love the diversity. I love the questions that I get to answer about the microbial communities. But I also just love the fact that nothing would live or survive without microbial communities, and myofauna depends on microbial communities. Macrofauna depend on microbial communities.
They're all
and in our deep
interconnected.
sea, everything eventually will make it to the seafloor, and so understanding how our ecosystems are functioning on our seafloor is super critical for the entire health of the ocean overall. And so I think it's just really cool to see sort of the base of that chain.
Amazing. I feel like y'all have so many like secrets about the really cool stuff nobody else knows about the seafloor. And I know you kind of just touched on that. But you mentioned that mud's super important to the ecosystem. So I also heard something about it helping with nutrient cycling. And so I wanted to just give you an opportunity to mention any other kind of fun facts about why sediment's important for the ocean, and then we'll thank everybody and head on our merry way.
Yeah, so microbial communities, they're kind of really a lot of your base of the nutrient cycles. A lot of your nutrient cycles could not occur without bacteria or archaea and other microbes. And so that's a lot of how myofauna and macrofauna and all the way up your food webs actually really depend on the microbial communities, is because they're the ones actually processing a lot of all of those nutrients into a form that's actually available to other organisms. And so, like I said, nothing would live without microbes.
Amazing. Well, I want to, oh, yeah, go ahead. Do you have anything else to add?
Oh no, just basically confirming what she said, and then also meiofauna is very important for the energy flow into the system, and then another important, I think, role that the meiofauna have is just the fact that all the meiofauna organism can move through the sediment, and this micro movement that is called microbiota can actually enhance the movement of the nutrients and oxygen through the sediments. That's another thing. For the microphone, I think it's the same.
It's the same for the macrofauna too.
Very important. And I want to thank all of you for taking time today to hang out with me and answer some questions from the audience and continue to grow my appreciation for mud. So thank you for your time. And we'll let y'all get back to it and get to your ice cream freezer or whatever else is next here on the docket.
It's time actually to deploy our megacore.
Yeah, we're gonna deploy. So.
Megacore. I love the phrase megacore. That's like a really good band name, actually. All right, so we'll let y'all go, and then I'll just share some updates with folks that are tuning in. We do have some live streams coming up next week and beyond. So if you like today's event, please tell folks you know and check out upcoming events from the University of Rhode Island's Inner Space Center YouTube page. The link is in the chat right now. And if you're watching a recording, then you are already here. Look for the little live icon where you can see what's coming up next. To learn more about these projects, find articles, publications, our story map, and hear about updates, check out the link that's in the chat. And also, we would love to get your feedback on today's program. We mentioned it at the beginning of the program, but if you did not get to that survey yet, please fill out that quick survey. That's also linked in the chat if you want to open it up on your browser to submit it after we wrap up. Please feel free. We'd love to hear from you. So thanks so much, and see you next time.